So let's sit down and we can get on with this. So you guys are real troopers. This is the last talk of the day. It's been a very long day, but this is the one of the. Huh? Yell? What? Okay. All right. So this is the last talk of what's been a very long day. Thank all of you for hanging in. I think this will be one of the well, how can I rate them? They're all fantastically interesting, but this is a mycological mystery that our friends Bryn Dentinger and Colin Domnauer, who you earlier heard his report on the Anagenanthera iconography. And now they're working together. Colin is a student, and Bryn is the curator of mycology at the Natural History Museum of Utah and the associate professor in the School of Biological Sciences at the University of Utah. Bryn received his BA from the McAllister College in St. Paul, my old stomping grounds, in 2000, and his PhD from the University of Minnesota in 2007. From 2010 to 2016, he worked as a senior scientist and head of mycology at the Royal Botanical Gardens, Kew in London. Not bad. And they are going to talk about these puzzling reports about psychoactive boletes. So, gentlemen, welcome.
Thank you.
Tell us how the boar ate the cabbage.
Thank you, Dennis. Hello, friends. Thanks for showing up tonight. I know it's been a long day. I know it's late.
Wouldn't miss it.
Yeah, let's like have a party. So we're talking about this mystery in the literature, and I think this illustrates a common problem in science, which is when there's something unknown, but there's a narrative out there, it's really hard to counter it without a counter-narrative to refer to. And so this is our sort of beginning to sort of unravel this weird mystery that may end up not being anything at all. And we'd love your feedback, I think, is what I'd like to say at the end. We'd love your thoughts and ideas about what might or might not be happening here. So I thought I'd start by first sort of laying the groundwork. I'm guessing most of you are not that familiar with mushrooms, mushroom taxonomy, diversity, morphology, and so I wanted to explain what the term bolete is being used to refer to in this case. And the term bolete, oh, there's a laser pointer, right? Yep.
It's okay.
Okay, I'll just walk. The term bolete refers to mushrooms that, so typical mushrooms that have a cap and a stalk, that instead of underneath the cap, instead of having gills, radially arranged blades of tissue on which the spores are produced, they have really tightly packed vertical tubes so that when you turn it over, it looks like a sponge. And it's inside these tubes that the spores are produced, just like they would be produced on the sides of the gills. And this is what you would find in your typical grocery store mushroom, or like the flammulina, the winter mushroom, Inokitake, that we had for lunch today would have had these, right? Okay, so that's the basic feature that distinguishes boletes from almost all other fungi. Mushrooms, I should say, sorry. So some cool facts about this group. This family, Boletaceae, is found everywhere in the world other than Antarctica. That's the only place they haven't been found. They're really rich in species. There's about 1,000 plus known species, but many more that haven't been described. That's a common theme in mycology, where the vast majority of the diversity has just never been documented or described. And that's true even for these, which I would argue are like the birds of the fungi. So these are probably the best known because they're very charismatic. Many of them are really good edibles, and yet we still only know a very small fraction of what's out there. They're really diverse morphologically, and this is an example of that. So I just told you a second ago that they're best known by their the pores that are produced, and you can't really see that from where you're sitting, but there have been guild versions that have been evolved from them, as well as truffles, including this really weird truffle from Malaysia, known as, Southeast Asia, known as Spongiforma. That species is close to Spongiforma squarepantsii. If you're a fan of cartoons, you know it's named after SpongeBob SquarePants. Most of these have a really specialized ecology, known as the ectomycorrhizal ecology. That's I'm abbreviating that with EM. This is where they live in a very intimate symbiosis with mostly woody plants, trees, where they exchange nutrients in the soil for sugar that the plants are making through photosynthesis. And this is a ubiquitous symbiosis. It's been estimated that over 60% of all woody plant stems globally form an ectomycorrhizal symbiosis. So this is hugely important ecologically. And these guys are almost all ectomycorrhizal symbionts. And not only that, they're among the top 3 most diverse and abundant in all habitats surveyed. So these are really important ecologically. They also have a really broad ecological amplitude. That means you can find them in really arid environments, you can find them in really wet environments, you can find them at sea level, you can find them at subalpine and subarctic habitats. So they span the whole range. So they're really an interesting group to study ecologically and evolutionarily. And then as a sort of added benefit, the icing on the cake is some of these are really economically valuable. Many of you in this room might have heard of porcini. Yeah, so porcini is in this group, and that's estimated to be worth over $100 million annually as a wild collected edible mushroom. Okay, so this group has traditionally been identified and classified using morphological traits of the mushrooms. And this is kind of a schematic sort of illustrating those. Some of the traits that have been emphasized include the spores that they produce. You can see that the morphology of the spores is quite varied, and they're quite beautiful under a scanning electron microscope, excuse me. And then there's some anatomical features. If you were to take very fine sections and look at them under a microscope of the tubes, you would see this in cross-section where the tube is made out of these parallel filaments of cells that then turn out and produce the fertile cells that produce the spores on the edges of the tubes. And there's 2 major arrangements of that tube called phyloporoid and boletoid. And this character has been used a lot for classification, as well as the presence of these sterile cells that stick out and have an unknown function as yet. The shape and size and color of those are also important, or have been traditionally. And then it turns out they also have been classified based on their secondary metabolite or specialized metabolite chemistry. So these mushrooms are known to produce really interesting compounds based on some of these core structures here. Oh, I guess I'll get to a little bit more elaboration on that in a second. One other thing about them that I think is relevant to the rest of the talk is that they're really variable. So from one mushroom to the next, you can see a lot of variation. And what I'm showing you here is the variation found in one species. This is actually porcini. So you can see there's quite a bit of difference between all of these. And we know from my work in my lab that these are not cryptic taxa. This is not bad taxonomy. That in fact, this represents the range of morphological variation of a population that has gene flow across it. They're also highly convergent. And so I thought this was a good illustration of an example of convergence in the group. This is Boletus variopes. It belongs to the porcini group. This is Tylopolis phelleus, the bitter bolete, which does not land anywhere near it evolutionarily. They last shared a common ancestor probably over 50 million years ago. And so the combination of high amounts of variation that exceed the variation you see between species makes it really hard to figure out what these things are. Okay. So we're interested in chemistry in this group because of the potential reports that we haven't gotten to yet. So I thought I'd just lay some groundwork here. We do know that they produce sesquiterpene molecules. This is a class of molecules that can have a lot of bioactivity. They can produce indole alkaloids. I think we've spent a lot of time over the last 48 hours talking about indole alkaloids. They can produce these nucleosides. This is an example of a nucleoside from Xerocomus nigromaculatus. And interestingly, this nucleoside is a synthetic anti-cancer drug that was unknown from nature until it was discovered in this mushroom. And I think this is a good illustration of just how profoundly important fungi are for chemistry. Fungi are chemical wizards. They are where we will look for the next anti-cancer drugs, the next antibiotics, the next therapeutics for all kinds of our ailments. They really are an underexploited resource. And then there are also some phenylethylamine and tryptamine molecules that have been identified in the boletes. And if you're not already familiar, phenylethylamine, a famous phenylethylamine that's relevant to our conversation today is mescaline from peyote and psilocybin from psilocybe and other mushrooms. Okay, so I mentioned previously that their specialized metabolites have been used to classify them, and a lot of these are forming pigment compounds. So they're really well known for these pigments that are derived from these basic molecules here. So yellow pulvinic acid is a major core molecule, and then the oxidation of this results in a bluing reaction, and I'll show you that in a second here. So these derivatives are often manifested as yellow to red, yellow, orange, and red pigments. And so this is a really common theme that has evolved many times throughout the family as they evolve these enzymatic capacities to modify these base molecules. And then when they get oxidized, do I just press the next button? We'll see. Yes. They can turn instantly blue in the presence of oxygen. And this is sometimes a spontaneous reaction, and sometimes it's an enzymatically catalyzed reaction. Okay. You could watch that in a loop over and over, right?
Did you take something?
Okay. What's that?
Did you take something?
Me? No. Not yet.
So with that background set, where do these reports of psychoactivity first come from? Well, it originates back in the early 1930s with the first contact in Papua New Guinea. Early missionaries noted that their encounters with the native peoples there, that they were seen to be sometimes overtaken by this mushroom madness, they called it, which was characterized by running amok or acting outside of the social norms of the society. And this quote here, he says, "The wild mushroom called Nanda makes the user temporarily insane." So this was the first missionary to contact these people, and already there are reduced reports of these mushrooms. Later in the 1950s, anthropologist Marie Rie starts living with the Kuma people in the Papua New Guinea highlands, and she notes this phenomenon in much more detail. These photos she documents of this mushroom madness occurring, and again she references these effects of after consuming the mushroom they began to experience Lilliputian hallucinations, they're called, and characterized by seeing these small diminutive creatures surrounding one's environment and causing a disturbance. So what are Lilliputian hallucinations? Well, it's a clinical term, actually, that was coined in 1909 by French psychiatrist Raoul Leroy, and named, obviously, after Gulliver's Travels. And so it's characterized, it's a actual psychiatric condition that's characterized by the perception of these tiny human, animal, or sometimes these fantasy cartoon-like figures. And he said they were always 10 to 30 centimeters tall, which is kind of interesting how restricted that is. And he described them as being characterized by these features of being mobile, colorful, numerous, and abounding, like in the thousands to millions, even in people's visual fields, and always reported as being rendered in exquisite detail. So this quote is summarizing that. He says, they are often reported as appearing in great numbers, apparently inhabiting the patient's external world, marching under real doors, climbing on real chairs and tables, while generally respecting the laws of gravity and three-dimensional space. As such, they fall into the category of reality-grounded projective hallucinations. So this is from the clinical psychiatric literature. This has nothing to do with mushrooms right now, but this is the clinical definition of this symptom. And just last year, there's a great review article written by this Dutch psychiatrist where he compiles basically the last century of reports of Lilliputian hallucinations in the literature. He was able to gather 226 published cases, and he described that 97% of these were reality-grounded, three-dimensional, lifelike figures. And you can see the little arrow in his tables where he outlines the causes of this symptom. Most commonly, it's like 40% of the time it's invoked by alcohol withdrawal. But other causes include like central nervous system lesions or a whole suite of drugs, mushrooms potentially being one of them. So following Marie Rie's anthropological reports in the 1950s, this reached the era of Gordon Wasson and Roger Heim. They obviously were interested in this topic after just making the expeditions to Mexico to describe the psilocybe mushrooms just 5 years prior. So they decided to visit Papua New Guinea and see what they can find. Roger Heim, the French mycologist, was able to survey 400 mushroom species in the area, and he described 6 bolete species said to cause this mushroom madness as it was reported to him by the native people. One particular species, photographed here, he named Boletaceae manicus, suggesting the mushroom madness. He sent this sample of Boletaceae manicus to Albert Hofmann. Just 5 years prior, like I said, Heim supplied Wasson with this psilocybe mexicana from Mexico. Hofmann was able to isolate psilocybin. I presume he was hoping for a similar situation here, but alas, no such promising results. Hofmann was unable to identify any active compounds. More recently, in just 2006, there's an anthropological survey conducted of interviews with the local population to see what the state of affairs of this mushroom madness is now. And I guess, unfortunately, unsurprisingly, most knowledge on the subject has been lost, with people saying, oh, I remember, actually some of the older elders in the tribe said, oh, I remember Wasson and Heim coming here 40 years later, but most of the knowledge on the subject of mushroom madness and the consumption has faded away. The last incidence, they said, was in the 1980s. However, one old man reported his experience with the mushrooms in the past. He said, I saw tiny people with mushrooms around their faces. They were teasing me, and he was trying to chase them away. So it was concluded by Heim, Wasson, and Murray Ray, the anthropologists, that this, due to their inability to find any active chemicals and the lack of consistency in the reported effects, that it was actually just a cultural drama that they enacted, that this was not a pharmacological effect of active mushrooms, but rather an opportunity for social catharsis and acting out. Heim and Wasson concluded the mushrooms do not seem to cause physiological effects leading to badness. So that's where we were left in the 1960s. However, 5,000 kilometers away in Yunnan, China, in just the past few decades, similar reports of psychoactive boletes have begun to emerge, with strikingly similar descriptions of causing Lilliputian hallucinations. The earliest article I can find published on this case in China was from 1991, where these doctors from the Yunnan hospital analyzed 300 cases of patients coming into them who were said to have been poisoned by a bolete mushroom. And they described the symptoms of these patients as something coming on 6 to 24 hours after ingesting, while lasting days to weeks. They wrote that all of the poisoned patients complained of Lilliputian hallucinations and claimed they saw little people and animals moving about everywhere. They saw them on their clothes when they were dressing and saw them on their dishes when they were eating. The hallucinations were even more vivid when their eyes were closed. So this was what the doctors reported in the hospital, but they took it one step further, which I don't think you could do today, but they took extracts of this mushroom and administered it to animals as an enema. I don't know why. But they wrote that they fed it to dogs and to a monkey, actually. And they described abnormal behavior appearing 6 to 8 hours after the extract was administered. They stared with the look of inquiry and fright, and they barked and were unable to fall asleep. This could just be a reaction to getting an enema that had absolutely nothing on it. Similar explanation with the monkey. Perhaps the most popularized article of this phenomenon was by David Aurora in 2008. He reported in his travels to Yunnan of local people telling him that sometimes if you undercook these blue staining boletes, you'll see little people. These are quotes from his article, initially said by these people in Yunnan. One guy said, "I asked for stir-fried mushrooms, and just as I was getting ready to leave after eating them, I noticed moving colors and shapes. I lifted the tablecloth just a little and peeked under. Hundreds of xiaoren." This news article I found from China of this Yunnan woman intentionally poisoning herself with this bolete for 8 years in a row just to have hallucinations to see her dead daughter. Well, she went to the hospital every year, and these nurses would tell her to stop eating this mushroom, but she insisted she wanted to see her dead daughter, and she said, "Well, I didn't see my daughter again. Instead, I saw a lot of strange things."
That's me. Okay, so we're now going to enter the phase of total confusion. So as I started out telling you, these things are really hard to tell apart from each other. And so it's completely unclear at this point if there's one culprit involved or many. And as an example from the article, we have these quotes about They're called the blue in the hand mushroom. Of course, different people apply that name to different things depending on where you are or where they were collected. And that blue staining reaction, which I showed you that video of, is super common across these mushrooms. So that alone is not a diagnostic character. Again, also people tend to assign the psychoactivity to what appears to be a number of different species, all of which are blue staining. So, I mean, the consistency with the blue staining is sort of encouraging, right? But otherwise, it's really still totally unclear. And then David Aurora continues to say that he was dazzled by the variety of blue staining boletes for sale, nearly 20 species. That's just based on his morphological examination, so there could even be more. We're like switching slide to slide now, so.
And mushroom poisoning in general, not necessarily psychoactive, but this remains a huge problem in China. From 2010 to 2020, there were over 10,000 mushroom poisoning incidents in China, 40% in Yunnan. They're obviously a largely mycophilic culture. They love eating mushrooms and collecting them from the wild and rely on them as a major food source. But the China CDC just reported this year that more taxonomic work is definitely needed, and the challenge of diminishing this high incidence of poisoning is motivation for conducting further research.
So it seems like, unlike in westernized civilizations where there are very particular species that are eaten intentionally, in China it's different. I think people will eat just about anything, and there's cultural knowledge about how to prepare them properly so that you don't get poisoned very often. Obviously, it still happens. And just sort of as an example of how this issue is now become a global issue, my lab looked at some commercially available dried mushroom products, and we sequenced the contents to figure out what was actually in these. And this packet of porcini, which I was able to buy from a large international online retailer, who I will not name, it turns out that we found a lot of things in it, none of which was porcini. Not even close. Including a bunch of boletes, some of which we couldn't even identify to species, as well as this thing, Amanita pseudoporphyria, which has been documented to cause acute renal failure in humans. And when you look at the customer testimonials on this large online retailer's website, it's perhaps not a surprise that there's poisonous mushrooms being sold. I would say the quality control, there's room for improvement. So anyway, I mean, it's just complicating this whole quagmire, right? Like, are we even talking about mushrooms now? I don't know. Okay.
Yeah, so there's potentially many species involved. This is reported that species as diverse as in different genera of Boletaceae, from Butreboletus to Lanmaoa to Sutorius, have all been suggested as this species that can cause these symptoms. But the probability that these different genera all contain the same active chemicals is highly unlikely. It's more likely a problem of the taxonomy needing to be sorted out more clearly. And so these are the diverse species that have been implicated or written about as causing these psychiatric conditions. And as you can tell, they generally, I mean, yes, there's like surface differences, but generally at a glance they do have a certain resemblance to them. And so perhaps similar appearances may lead to confusion. Perhaps different species are being mixed together or even eaten together, even if they're able to identify them, but they all mix them together in a single meal. It's difficult to pin down the culprit. And so the China CDC in their annual report highlighted this fact that more research is needed to mushroom poisoning. And they specifically wrote about the species called Lanmaoa asiatica, and they discussed this problem that it can cause hallucinations, and they say this is a different thing than psilocybin, and its toxicity is still unclear and needs further studies. And this was just from a few months ago.
Okay, so assuming Lanmaoa is correct, maybe we can learn something about it. Maybe there's more information we can glean from the knowledge that's available that could lead us down one path or another to understand what's going on. So first of all, like, what is this genus? It's a recently segregated genus from Boletaceae. There's a lot of taxonomic upheaval at the moment. And it's defined by having these short tubes. That is the length of the tube. So a lot of the Boletaceae have much longer tubes than this. But again, like the blue staining, that's not a character that's unique to this genus. It's supposed to have a light yellow context that stains blue when exposed. Again, not a character unique to the genus. And then it's got a specialized anatomy of the cap surface that, again, is not unique to the genus. So you can maybe start to appreciate the problems we have with trying to figure out how things are related to one another in the Boletaceae and other mushrooms. One thing that I think is interesting and might be related, which hopefully will make sense in a few slides, is that many or all of the species in this group have this distinct, really savory odor. It's something that you can smell when you cut it open. In fact, some of the common names in like the US for this is the bouillon bolete, because when you cut it open, it smells like chicken stock. Or some people have described it as onion. This is the type species of the genus, and it's known only from these montane pine forests in Yunnan. And the holotype, that is the type that represents the name, the one physical voucher that represents the name, it was acquired from a market, which is not ideal. Because that means it's really hard for us to go back and try to reproduce this information should we need to do that, like, say, to confirm the chemistry or the species ID. However, interestingly, it turns out that this genus is not found only in Asia. And in fact, the closest free-living relative to it is in North America. For me, that is something that's potentially hiding in plain sight. So there are no reports of psychoactivity from any of these boletes in North America or Europe. In fact, nowhere outside of Papua New Guinea and China are there reports of psychoactivity of these mushrooms. But a lot of people don't eat these in North America and Europe because there is a cultural stigma against eating boletes that stain blue. So it's possible that we've just missed it because nobody ever does bioassays on themselves, right? And so this is kind of interesting and kind of an exciting opportunity because we have ready access to this species where I am and could do preliminary chemical analyses on it on the assumption that its close relationship predicts that it will have similar chemistry. Okay, so let's just pretend that this is the culprit and that we need to know what chemistry is involved. Okay, so given our current understanding of psychoactive compounds in mushrooms, there's really only two compounds. There's the ibotenic acid prodrug resulting in muscimol, and there's psilocybin prodrug resulting in psilocin. Interestingly, psilocybin the biosynthesis of psilocybin has evolved through horizontal gene transfer, meaning the genes involved in synthesizing psilocybin have been inherited through direct contact or through some vector rather than vertical inheritance, the normal type of genetic inheritance. This was once thought to be a really rare form of inheritance. It's now been documented more and more, and we're beginning to realize that horizontal gene transfer is a lot more common than we thought. So we think it's happened at least twice with the biosynthetic gene cluster for psilocybin. It also has evolved apparently convergently. So this species that we sequenced, which produces psilocybin, which we have been able to prove with chemical testing, does not have the biosynthetic gene cluster for psilocybin that's found in all the other psilocybin-producing mushrooms. This strongly suggests that this species has evolved the ability to produce psilocybin with a completely different set of genes. We're currently following up on that.
Would that resemble the biosynthetic pathway in Massospora? This strange pathogen on the cicadas?
Yeah, so Massospora is another really interesting possible independent origin of psilocybin biosynthesis. Right. No genes were found in the genome of Massospora that correspond to the known biosynthetic gene cluster. That's the current stage that we're at with this study. We are currently doing work to try to figure that out. Yeah.
Very interesting.
So when it comes to ibotenic acid, it turns out that the biosynthesis was just recently elucidated in Amanita muscaria. And there's a good chance, I would say, that it also has evolved either convergently or through horizontal gene transfer. And the reason for this is it's known that there's a species of tricholoma, which is not in this phylogenetic tree, that produces tricholomic acid. Tricholomic acid is the immediate precursor to ibotenic acid. So then we can pose the question, given that we know these are the two psychoactive compounds known for mushrooms, could we find the same genetic machinery in Lanmaoa that they have? So in the ibotenic acid biosynthetic route, it's derived from a hydroxylated glutamate. Interestingly, remember I said that these mushrooms have a strong chicken stock or onion odor. This is likely due to glutamate molecules. And a lot of mushrooms, the reason we like to eat them is because they provide umami, that rich, meaty, satisfying flavor. That's from the glutamates, including MSG. Monosodium glutamate, found in many mushrooms, in fact. It's a natural product. So that's an interesting connection. It's totally circumstantial, of course, but interesting to note. This mushroom, Lanmaoa asiatica, as well as I've read accounts, anecdotes of Amanita muscaria, is really delicious. It's got a lot of umami flavor that you want out of your mushroom. So maybe it's producing a glutamate-derived compound that is interacting with GABA receptors, and some of the symptoms are consistent with that. So here I'm showing you, this is the biosynthetic pathway. It starts with conversion of glutamate to 3-hydroxyglutamate using a gene known as IBO-H. And it goes through a couple of other steps, and then it ends with the conversion of tricolomic acid to ibotenic acid with a gene called IBO-C. I'm pointing those two out because I'm going to refer to them in a minute. Okay, so if we were lucky enough to have a genome, we might be able to mine the gene content of that genome and look for these enzymes. Well, it turns out there is a genome available. It is labeled as Lanmaoa asiatica, and I emphasize that labeled part. One problem is that the true identity is somewhat ambiguous, and that's because when I fished out the DNA barcode region, so the region of the genome that's used to identify species, it's different than the DNA barcode region from the only other sequence available for this species in the public databases. Unfortunately, neither of these come from the holotype specimen. So who knows what these are? But let's pretend it is Lanmaoa asiatica, because it's fun. Okay. So one cool feature about fungal biosynthetic genes, is that they tend to be clustered together physically. They're found together in the same region on a chromosome. That makes them relatively easy to search for bioinformatically, because you can ask, is there a gene annotated to perform this function that's physically close to a gene that's annotated to perform this other function? Okay, so this is a feature that has been used widely for sort of preliminary mining of biosynthetic gene clusters from fungi. In fact, there are 2 multi-million-dollar companies that have been founded solely on this principle, and they're doing great. Oh, but I should say, because the pharmaceutical industry has largely focused on Ascomycota, these are the molds in yeasts, the models that we have to identify these genes and figure out what they are based on our knowledge from Ascomycota. The mushrooms are from the Basidiomycota. They last shared a common ancestor with the Ascomycota about 600 million years ago. And so those models are actually really poor indicators of the genes and the functions in the mushrooms. Okay, so nonetheless, I forged ahead because I had something to say. I needed something to say. And I used a commonly used database called Fungi Smash or Anti-Smash. This was developed to identify antibiotic biosynthetic gene clusters in bacteria. It's been extended to the fungi. And in this genome of Lanmaoa asiatica, there were 15, I forgot now, something like 15 biosynthetic gene clusters predicted based on this, this software. They're really rich in these non-ribosomal peptide synthetases and terpene biosynthetic pathways. But there were no biosynthetic gene clusters predicted that were similar to known biosynthetic gene clusters. That means we can't tell what they're doing, really. We can just say at a very coarse level that they're probably involved in terpene biosynthesis. Okay, great. I could have told you that before doing this work, right? And the other problem with mining fungal genomes is because they're so poorly characterized in terms of the gene function, a lot of the returns on the annotation of these genes, so we can say that's definitely a gene or looks really like a gene. What kind of gene is it? The result, the answer back from your magic 8 cube is hypothetical. It's a hypothetical protein. We think it's making a protein, no idea what it does. And that's a real problem because we can't really go any further with this approach. So I took a slightly different approach. I thought, okay, if we know that there are 2 classes of psychoactive drugs for mushrooms, can I hone in on those 2 classes and figure out if there's a gene cluster or a gene that might be involved in the biosynthesis of them? So the first thing that I did is I fished out the Psy-D gene. This is a gene that's critical in the transformation of tryptophan to tryptamine in the psilocybin biosynthesis pathway. And I used an approach called reciprocal best blast. You don't need to know what that is. And then I built a phylogenetic tree out of the best hits from Lanmaoa asiatica and the other 2 genomes that my lab has generated that are related. I generated that in the presence of the same gene, the homolog, from the psilocybin-producing mushrooms, psilocybe, gymnopolis, paniolus, as well as that independent origin in the inocybe. And you can see that the genes, which are in red here from the Boletaceae, are not close relatives of either of those. That doesn't mean that it's not involved in producing a tryptamine derivative that could be psychoactive, but it means it definitely doesn't look like the ones we know about. Okay, I did the same thing for the ibotenic acid biosynthetic gene cluster, and I honed in on that first step, the IBO-H gene, which converts glutamate to 3-hydroxyglutamate, and the last step, IBO-C. I did them all, actually, but I didn't want to torture you. So I'm just showing you these two, which seemed most critical. And again, you'll just have to take my word for it, because I'm sure you can't read this. These genes, which are highlighted in yellow, which come from the Lanmaoa asiatica genome, are not anywhere related to the Amanita muscaria genes. Okay. So, you want to finish up, or should I just keep going? Okay. So, just in summary, where are we now and where are we going? So, there are these abundant reports of blue staining boletes in China that have these similar psychophysiological effects, reportedly. They're commonly eaten as food. Occasionally, there appears to be unintended poisoning that could be for a number of reasons, and many people are sent to the hospital. But the chemistry, toxicity, and taxonomic identity of the species remains totally obscure. We really need some hardcore, fundamental taxonomy done to really just start from a baseline. From there, we might be able to develop real hypotheses that can be tested about the possible chemistry involved. And then we can move on to these more sophisticated techniques, which we can apply if we know what to apply it to. And then I think this is really going to require working very closely with the people in Yunnan, because they are the ones that have this knowledge. And without being able to establish a good rapport and relationship with them, we're never going to get to the stuff on the top half of that slide. Okay, so let's say we were able to identify a potential culprit. We were able to establish its identity. There are still challenges, and I just wanted to share some of the lessons we've learned recently from our work with psilocybe in collaboration with Paul Stamets. And this is an example of how the chemistry is really sort of unstable, and that presents a whole nother layer of challenge here. These are the three best known, most highly collected species found in fungal herbaria, Fungaria. And what we did is we were able to quantify the amount of psilocybin, psilocin, and the intermediates here, as well as tryptophan in specimens over a range of ages. And we were surprised, as you might be, that there were almost no specimens of Psilocybe cubensis that rendered quantifiable content of psilocybin or psilocin. Except for this one here, which I believe is a specimen Paul deposited at the University of Washington in 1978.
Of course.
Congratulations, there's still psilocybin in your specimen, Paul. Other than that, it's undetectable, which suggests that psilocybin and psilocin degrades very rapidly in Psilocybe cubensis. That is not the case for these two species, which is really interesting that there appears to be a taxonomic pattern to the stability of these compounds. It's not directly age related, right? There's obviously other factors involved, but there is an age component to those patterns.
Is that bottom graph years? Is that the horizontal axis years?
Years, yes. Yep.
And how many years? I can't see it.
So the oldest specimen we have is just about 50 years old. The other thing is, I think this was mentioned in the talk on kratom or kratom, or I don't know how to pronounce it. That one. It's easy to hone in on the one molecule that we think is interesting, and then we lose sight of all the other potential stuff that's going on. And this is true with psilocybe and psilocybin as well. And we were interested to know what other kinds of compounds might be present in psilocybe mushrooms. And so we took a non-targeted metabolomic approach. We grew Psilocybe cubensis, the same strain, same genetic strain, and then we harvested it in 2 flushes in one bin, and then the same first flush from another bin, because we wanted to know bin-to-bin variation and within-bin variation. And then we divided it anatomically between cap and stipe, and then we also analyzed mycelium. And what we found was that, so these are the compounds that we found. These are clustered according to their spectral features. I think one of the main take-homes here is we found just about 1,500 different spectral features. Now, these are not equivalent to compounds, but they're pretty close. So there's a lot of chemical diversity that we don't know anything about. I think that's the main message here. The other message is that there's remarkable consistency in the chemical profile between flushes and between bins. So they were grown, same strain, grown under the same conditions on the same substrate. Okay, so those were held constant. There's actually quite a bit of stability here in the chemical profile. I think that's kind of important to know. Anyway, this kind of information just sort of adds complexity to our search for the possible chemical basis of this weird mystery psychoactive bolete in China. Okay, so I wanted to end with some poetry. This is one of my favorite poems by William Carlos Williams. And it says, there is the microscopic anatomy of the whale. This is reassuring. Among other things, the reason that I put it here is this tells us that we know a lot about very little. And this reflects something Alfred Russel Wallace said in the Malay Archipelago, which is that we should not be drawing general conclusions from a very partial view of nature. And so when we only know 5%, maybe 10% of fungal diversity, I think we really need to focus our resources on filling in that gap before we can really hope to understand these weird mysteries. You want to add anything?
That was great.
Okay. A lot of people to thank here, but first and foremost, ESPD 55, the McKenna Academy, and especially Dennis McKenna for inviting us, and Annette for her amazing ability to herd us crazy cats. So thank you all. It's a real honor to be here. And then there are some collaborators on the Psilocybe project, people that provided specimens and help with some of the bioinformatics. And the work on the Boletaceae is supported by a grant from NSF, and so I want to acknowledge them. Okay.
Okay, we'll open the floor to questions.
Thank you both for a really beautiful, beautiful talk. And I would like to offer an appreciation and ask a question and register a concern. My appreciation is that this is the most amazing way of narrativizing a deep fungal nerd hole and using the best possible methods that we have right now with all of the latest technologies and thinkings and philosophies and engineerings, and you two work so well together, and I appreciate so much what you brought tonight on lots of levels and in a big way. My question is, do you think that the phylogeny of fungi, given the fact that we know from what you said and from the studies that have proved it, that the psilocybin gene cluster moved horizontally between different lineages, do you think, given the evidence of horizontal gene transfer in fungi, that taxonomic vertical lineages can really tell us that much about what everyone's doing metabolically? And my concern is that, given that the reason why you're investigating this bolete is because there's lots of phenomenological examples and ethnographic examples and self-reported examples. My concern is there haven't been enough bioassays in your experiment, i.e. you two going out there and having some soup that's been undercooked. Do you not think we'd get a little bit quicker to the question about whether these things were working or not if you just took the fucking mushrooms?
Well, first of all,
It's unclear if they're safe to consume, first of all. I mean, we don't know. Maybe they're also, in addition to being psychoactive, toxic. In China, everyone who gets poisoned and sees little people, they immediately send themselves to the hospital. Whether that's a necessary precaution to take or not, or we don't know. But I would tread lightly.
of course, you don't gain anything by being risk-free, right?
What about the question?
Wait. I'll just let me add one thing, which is, to heighten concern over this approach. It's recently been shown that the gene cluster that produces amatoxins, which are the most notorious toxins produced by mushrooms, they're produced by the death cap mushroom. You're almost guaranteed certain death if you eat them. Those have been shown recently to also have been acquired horizontally.
So now answer the question. Now answer the question. You deepened the question.
About vertical gene flow.
Oh, yeah. Oh, so, yeah, I mean, yeah, I don't want to, undermine my justification for what I do, which is that phylogenies are useful for predicting functions. But I do wonder, actually, how rampant horizontal gene transfer is when it comes to the metabolic functions of fungi that determine their ecologies. So I guess I'm punting this a little bit.
I appreciate your honesty.
Okay. I don't think I'm going to go any further without a glass of wine.
All right, next question.
Yeah, just for myself and everybody here, is horizontal gene transfer occurring via mycelial contact directly?
We have no idea.
How else could it be?
Could be viral.
Oh, interesting. Okay, viral.
I would say we've looked at the possibility of viral transfer in the psilocybin biosynthetic gene cluster, and the genetic signatures are not consistent. So viruses don't have introns in their genes, and there's no indication. The intron structure is the same. So that suggests that there was not a viral vector. We think our current thinking, and this is not even a hypothesis, is that it is through direct contact. So it's been shown that filamentous fungi can have what are known as supernumerary chromosomes. These are similar to plasmids in bacteria that might move freely between cells. Total guess. But that's my current thinking. The one thing that sort of is consistent with that is the pattern seems to be associated with ecology. So the psilocybe that grow on wood chips or downed wood, that cluster is the same cluster you find in the wood-dwelling Gymnopolis in Pluteus.
Could it be endobacteria that are being exchanged?
Yeah, we don't see a shift in codon bias, which you would expect if it went through a bacterium.
What about insect feeders?
Yeah, that's also a definite, I think maybe most likely vector. That doesn't exclude it being a supernumerary chromosome that's being moved, but yeah. Yeah, I will say we, just as a fanciful idea, I've had my student co-culture Pleurotus with psilocybe, and we're going to look for the genes in the Pleurotus, but we haven't done that analysis yet.
So it sounded like the SID gene decarboxylated tryptophan to tryptamine. Was it related to an aromatic amino acid decarboxylase or dope
It is
decarboxylase?
one, yes. Yep.
Thank you both. Are there any reports of Lilliputian hallucinations with intoxication from mushrooms or fungi that don't have psychedelic compounds, just basically being poisoned?
Not from mushrooms, but from a large variety of other psychoactive compounds, yes.
Always psychoactive. So Lilliputian is exclusive to psychoactive, or is it also associated to intoxication? No questions.
I'm confused.
Non-psychedelic intoxication.
Oh,
If it's hallucination, it's psychoactive, isn't it? I'm not sure.
Well, what I'm saying is, so, I've seen my son hallucinate from high fever, right? So what I want to say is there any systematic report of this type of hallucination
yes.
from anything other than a psychedelic?
Oh, yes. So 40% or like 40% were from alcohol withdrawal. Another 15% were from CNS lesions, and then I forget the number, maybe 20% was from ocular degeneration and age, Charles-Bonnet syndrome, that sort of thing. So yes, there's some underlying neurological mechanism that neurologists still don't understand the mechanisms of how Lilliputian hallucinations are induced, but it's interesting that potentially a compound from this mushroom could induce those effects reliably, which may give us some insight into this neurological condition.
Don't rain on our parade.
Sorry.
I think Juliana's question goes right to the heart of it. You have to look at phenomenology, right? Yeah, I mean, are there other plants perhaps that induce these Lilliputian hallucinations? Are there other, there are defined neurological conditions. So if you look at the pharmacological basis for that, the gene, characterization of ibotenic acid and psilocybin gene clusters in these Boletaceae is interesting, but pharmacologically it's off the mark because they don't, abotenic acid are GABA agonist, psilocybin is serotonin agonist, and it's not typical of the phenomenology of this Lilliputian hallucination. So, which is, again, I guess it's not always part of it. I think you have to look for, that's the raw datum. That's you have to find somebody that, that has these, that can confirm these Lilliputian hallucinations.
Yeah.
Who has basically a Western mindset, because the Chinese seem to have a different way of looking at it. But I don't know if there were another. You've looked at the literature on Lilliputian hallucinations. It's not always alcohol. You say neurodegeneration, ocular diseases. I don't know. I would look there. I would look there first, maybe, before looking for the genetic basis of it. But it's just an idea.
I completely agree.
Ethnopharmacology.
Yeah.
Sorry, just want to say that at the beginning of the talk, you asked for suggestions and ways forward.
Yeah,
That was my
no, this is great.
Second thing, could you explain why we haven't gone back to Papua New Guinea to look for the original account? What's stopping the field work?
They have gone back and, first of all, the cultural knowledge is gone, and then also much of the forest is gone from deforestation and drying up, climate change. There's a lot less diversity of boletes being fruiting there now. I'll leave it at that.
On a practical level, it's super hard to work there.
So okay, go ahead.
She asked why.
Logistically, it's hard to get in contact with people that can facilitate permits and getting into the field in the right places. And two, it's like super dangerous.
You mean in Yunnan or in Papua?
No, Papua New Guinea.
But Yunnan is where this phenomenon is happening regularly. People are going to the hospital with these.
Well, that's exactly my point. If the chain of custody is so ambiguous going into the past, what about going into the future? Why don't you contact the CDC in China and say that we're interested in the pharmacology, and when people come in with the symptoms, have them save the mushrooms so you can get voucher specimens and then be able to track this.
That would be a start.
That would be, and they would have a vested interest in that, and that would be a collaborative. It seemed like a logical call out that they've already identified it as an issue.
That's a great idea. I should point out that Colin started as a PhD student this August.
So can we get a plane ticket together for Colin to head over there?
Yeah.
Serious.
Okay, thank you. We're going to take one more question from Glenn. Yeah.
Forget you're a geneticist, mycologist, whatever. Let's just bet $100. What does your gut tell you? You're going to bet $100. There's some unknown psychoactive bolete with something or not. What's your bet? I'm going to bet with the indigenous people of Yunnan and the indigenous people of this totally different region, New Guinea, which is an incredibly similar thing about boletes and seeing little people, blah, blah, blah. Yunnan, New Guinea, it's not like they're next door neighbors. I'm going to bet with the indigenous people against these biologists with all these, genes. That's one or two things. What's your bet?
If we were in Yunnan, this wouldn't be a debate. It's an accepted fact there. So yes, I agree.
I want to hear from
What
him.
are you going to bet, Colin?
I'll bet Paul's $1,000.
I want badly for it to be a real thing. And so I'm going to operate under the assumption that it is a real thing that needs to be figured out until I have been beaten in the head so many times that it's not.
So what are you going to bet?
Oh, yeah, I'll bet you $200.
Okay.